Hello and welcome. Today we will focus on Illumina sequencing by synthesis, also known as SBS. In the previous lecture, we discussed 16S rRNA Amplicon sequencing for analyzing microbial communities. Before going into bioinformatics analysis, we have to understand sequencing techniques to better understand and interpret the data. Now, to understand how those sequences are actually generated, we need to explore the sequencing technology behind it. There are several sequencing technologies available today. Some of the major ones include Illumina sequencing by synthesis or SBS, Pacific Biosciences single molecule real-time sequencing or SMRT sequencing, Oxford Nanopore technologies, that is nanopore sequencing, and earlier technologies like Sanger sequencing and Pyro sequencing. Among these, Illumina sequencing by synthesis is one of the most widely used technologies in modern genomics. And this is what we will focus on today. So how does Illumina sequencing actually work? Let's break it down step by step. The core idea behind Illumina sequencing is simple. DNA is sequenced one base at a time by detecting the incorporation of nucleotides. This process is called sequencing by synthesis. In simple terms, we copy the DNA strand and read each base when it is added. Here are the steps in short. First is library preparation, where adapters are added to the ends of the DNA fragments to be sequenced. Next is cluster generation by bridge amplification, where DNA library fragments hybridize to complementary oligos on the flow cell surface and are then amplified through bridge amplification to generate clusters of identical DNA copies. Then sequencing, where the instrument incorporates nucleotides into the growing DNA strand and records the growing chain of DNA sequence one base at a time. Finally, primary data analysis begins. This is where raw imaging data in the form of TIFF files are processed and converted into CIF files containing intensities, and then into BCL files containing base calls and quality scores. These BCL files are then converted into FASTQ files for downstream bioinformatics analysis. We will go into details for each of the four steps of the sequencing workflow. First is library preparation. Library preparation is the first and one of the most critical steps in Illumina sequencing. In this step, genomic DNA is converted into a form that is compatible with the sequencing platform. Before sequencing, DNA must be prepared into a sequencing library. First, DNA extraction is performed. High quality DNA is isolated from the sample of interest. This sample can come from a wide range of biological sources. This may include clinical samples, environmental samples, or the human microbiome. It can also come from any type of organism such as bacteria, viruses, or eukaryotes. The quality and purity of DNA are very important because poor quality DNA can affect downstream analysis and results. Next, DNA fragmentation. The extracted DNA is fragmented into smaller pieces, typically ranging from 200 to 500 base pairs. This fragmentation can be done using mechanical or physical methods like sonication or enzymatic methods using specific enzymes. After fragmentation, the DNA ends are processed. This step is called end repair. During end repair, overhangs are removed and blunt ends are generated. Then a single A base, that is adenine, is added to the 3' ends of the DNA fragments. This step is called A-tailing and it helps in the efficient ligation of the adapters. Now comes a very important step, adapter ligation. Short synthetic DNA sequences called adapters are attached to both ends of each DNA fragment, resulting in a complete library. After adapter ligation, the DNA library is often purified. This removes unligated adapters, small fragments, and contaminants. Next, PCR amplification. The adapter ligated DNA fragments or library pool are amplified using PCR or polymerase chain reaction. This step increases the quantity of DNA or libraries and ensure that there are enough material for sequencing. However, excessive PCR cycles should be avoided because they can introduce bias. At the end, a complete library structure is formed. Each DNA fragment becomes a complete library molecule ready for sequencing. Let's take a look at the full structure of a library molecule. Each complete library will have the DNA insert which is the target fragment of the double-stranded original DNA piece extracted from the sample. This is the region that will actually be sequenced. Next, two unique adapter sequences known as P5 and P7 adapters. These are added to the both ends of the DNA fragment. These adapters contain the binding regions for clustering, that is P5 and P7, sequencing primer binding sites, and the index sequences. As we said, these adapters include the binding regions for clustering, short synthetic DNA sequences called P5 and P7, which are complementary to the flow cell oligos, and these facilitate library hybridization to the flow cell. One end binds to P5 oligos and the other to P7 oligos on the flow cell surface. The adapters also contain sequencing primer binding site, which are used to initiate sequencing reactions, including read 1 and read 2 primer in paired end sequencing. There is a read 1 sequencing primer binding site for the forward read and a read 2 sequencing primer binding site for the reverse read. A single read library would only be read in the forward direction, and a paired end library would be read in both forward and reverse directions to increase coverage. Lastly, the index sequences, also called barcodes, are present in the adapters. Indexes such as I5 and I7 are short DNA sequences, normally about 6 to 10 base pairs long, that act as barcodes to uniquely identify samples within a sequencing run. The I5 index is located on one adapter, while the I7 index is on the other adapter. Index sequences are important because they allow multiple samples to be sequenced together in a single run and later separated during data analysis. This process is called multiplexing. In this example, two indexes are present, signifying a dual index library. If there is only one index, the library would be a single index library. Now let's move to the next step, flow cell and cluster generation. After library preparation, the sequencing library is loaded onto a flow cell. A flow cell is a glass slide with multiple lanes coated with millions of short single-stranded DNA oligonucleotides. These oligos are complementary to the adapter sequences P5 and P7 present at either end of the library fragments and allow the library to bind to the flow cell. So, first, double-stranded library fragments are denatured to form single-stranded DNA molecules. These single-stranded fragments are then loaded onto the flow cell through each lane. Each fragment binds via its adapter to a complementary oligo on the flow cell surface. At this stage, each fragment is immobilized and ready for amplification. The next step in Illumina sequencing is cluster generation by a process called bridge amplification, where the prepared libraries are amplified on the flow cell and form clusters that will be imaged during sequencing. So what is a cluster? A cluster is a group of thousands of clonal copies of the same DNA library fragment located in very close proximity on the flow cell. These closely packed copies are required to produce sufficient signal intensity for detection by the optical system during sequencing. Each cluster begins as a single DNA molecule. It is then amplified to produce many identical copies. This is important because it increases the signal intensity. Without cluster generation, the signal from a single molecule would be too weak to detect. During the sequencing run, all strands within each cluster are imaged in every cycle. Each bright spot represents a cluster producing signal. We will show it later. Dark regions indicate areas where no clusters are present. The instrument uses these images to determine cluster location and signal intensity, and to identify which base is incorporated at each cycle. Now we will discuss cluster generation in details. First is the extension. The immobilized library fragments are then primed from the flow cell oligos. Here, priming means that the flow cell oligo acts as the starting point for DNA synthesis during cluster formation. It provides a free 3' hydroxyl group required for DNA polymerase to initiate strand extension. A DNA polymerase extends a complementary strand using the hybridized fragment as a template. After extension from the flow cell oligo, the library becomes double-stranded, including both the original template strand and the newly synthesized strand. This newly synthesized strand is covalently attached to the flow cell via the oligo linkage. This completes the first strand synthesis step. Next, the double-stranded DNA is denatured and the original template strand is washed away. This leaves only the newly synthesized single-stranded DNA anchored to the flow cell. Now begins bridge amplification. On the flow cell, bridge amplification begins when each immobilized single-stranded DNA fragment bends over and hybridizes to neighboring complementary oligo on the flow cell surface. This creates a bridge-like structure between the attached strand and the surface oligo. Then the DNA strand is primed from the oligo similar to the previous extension step. A new complementary strand is synthesized. The DNA polymerase extends the bound oligo using the original fragment as a template while still in the bridge position. This results in the formation of a double-stranded DNA bridge. Then this double-stranded bridge is denatured. So there are two single-stranded templates both attached to the flow cell surface covalently. One strand remains anchored to the original position while the newly synthesized strand is also fixed nearby. So this process of bridge amplification doubles the number of library molecules on the flow cell and generates both forward and reverse strand. This process is then repeated during the next cycle of bridge amplification. These single-stranded DNA molecules bend over once again and hybridize to nearby complementary oligos. Now both strands can act as templates for amplification. They are then extended by the DNA polymerase. This results in the formation of two double-stranded DNA bridges. These double-stranded DNA bridges are denatured again. This doubles the number of DNA templates, resulting in 4 single-stranded DNA templates covalently attached to the flow cell. 2 of these strands are in the forward direction and the other 2 are in the reverse direction. This cycle is repeated multiple times. Each round of amplification doubles the number of DNA copies. Over time, this leads to the formation of dense clusters. Once the amplification is complete, the bridges are denatured, resulting in only single-stranded DNA anchored to the flow cell. Each cluster, localized at a spot on the flow cell surface, contains thousands of identical copies of the original DNA fragment. This is how a single DNA molecule is amplified to form a cluster. The amplification process is known as bridge amplification as it forms bridges. At the end of this step, we have millions of clusters, each representing a single original DNA fragment uniformly distributed across the flow cell and ready for sequencing by synthesis. Here is the summary of the cluster generation by bridge amplification. Cluster generation starts from a single DNA fragment. Then the binding fragment binds to the complementary oligos on the flow cell surface via adapter 1. Then the bridge formation. The fragment bends over and the other end binds to the nearby oligos. Then the extension. DNA polymerase extends from the bound primer to form double-stranded DNA. Next is denaturation. The double strand is denatured, leaving 2 single strands anchored to the flow cell. After that, the second bridge formation. The anchored strands bend over and bind to new oligos, forming second bridge. Next is extension and denaturation again. Extensions create double strands and denaturation leaves multiple anchored single strand. Finally, the cluster formation. Repeated cycles generate a dense cluster of clonal DNA fragments originating from the single original fragment. The final result is a cluster of thousands of copies from a single DNA fragment. After the cluster generation is complete, the DNA within each cluster must be prepared for sequencing. At this stage, cluster contain both forward and reverse strand, and the libraries are attached to both the P5 and P7 oligos on the flow cell surface. However, sequencing requires a single uniform strand orientation for accurate sequencing. Therefore, all strands within a cluster must be in the same orientation. So, one strand of the double-stranded DNA is selectively clipped and removed or washed away. This process is called linearization. This leaves only one type of strand attached to the flow cell. All remaining strands are now in the same orientation, allowing the instrument to correctly identify each base during sequencing. Next, the 3' ends of the flow cell oligos are blocked. This prevents DNA template strands from rebridging again or further extension from the 3' ends of the flow cell oligos during sequencing preparation. As a result, only forward strands remain on the flow cell and read one is ready to be sequenced. Now comes the main step, sequencing by synthesis or SBS begins. This is the core step where the actual DNA sequence is read one base at a time. Now the sequencing primer is introduced. The primer binds to the specific primer binding site present in the adapter region. For read 1, the read 1 sequencing primer binds to its complementary site. This defines the starting point for sequencing in the direction towards the flow cell surface. The key point is that the primer provides a free 3' hydroxyl group, which is essential for DNA polymerase to initiate strand extension. During sequencing, individual bases are incorporated into the growing DNA strand and each incorporation is imaged. Now the read 1 is complete. The first sequencing read has been generated. We will dive into the sequencing by synthesis in detail right after we complete paired end sequencing. Although sequencing from one end is complete, however, Illumina platforms often use paired end sequencing. This means DNA is sequenced from both ends of the fragment, so we obtain information from both ends of the DNA fragment. This is called paired end sequencing. This allows sequencing of the same DNA fragment from the opposite direction, generating read 2. This improves accuracy, alignment, and detection of structural variations. After all cycles of read 1 are completed, the newly synthesized read 1 strand is removed. The read 1 primer is also must be removed. So read 1 primers are washed away. This leaves only the original template strand still attached to the flow cell. Then the 3' ends are blocked again to prevent unwanted extension. Next, the template is reoriented and regenerated. Similar to the cluster generation, the strand bends over and hybridizes to a nearby flow cell. A bridge is formed again and the opposite strand is synthesized. The DNA polymerase extends the strand, forming a double-stranded bridge. This creates a complementary copy of the original fragment. This double-stranded DNA is then denatured, resulting in two single-stranded strands both anchored to the flow cell. These are now reverse complement-oriented copies of each other. Next, one strand is selectively clipped and washed away or removed, leaving only the strand required for read 2 sequencing. Now the flow cell contains the opposite strand orientation. Again, 3' ends are blocked to prevent rebridging. At this point, the read 2 sequencing primer is introduced. The read 2 primer binds to its specific primer binding site present on the adapter sequence. This defines the starting point for sequencing from the opposite end of the DNA fragment. Now the sequencing by synthesis begins again. Nucleotides are incorporated cycle by cycle just like read 1. Each cycle includes incorporation, washing, imaging, and base calling. Following the same sequencing by synthesis chemistry as read 1, this generates read 2, which sequences the DNA fragment from the opposite direction. Now let's take a closer look at sequencing by synthesis and how imaging and detection work. The template strand is shown here as a DNA molecule and the short sequence represents the sequencing primer. In Illumina sequencing, 4 types of fluorescently labeled nucleotides are used. In each cycle, a mixture of all 4 fluorescently labeled nucleotides is flowed over the flow cell. Here, red represents thymine, blue represents adenine, yellow represents cytosine, and green represents guanine. They also contain a reversible terminator. This terminator ensures that only one base is incorporated per cycle. Each nucleotide carries a unique fluorescent dye corresponding to A, T, G, or C, producing a unique signal when excited by a light source. During each cycle, nucleotides are incorporated into the growing strand. Each cluster incorporates only one complementary nucleotide based on the template sequence. Once a nucleotide is added, unincorporated nucleotides are washed away. Then the flow cell is imaged. The instrument then pauses and a light source excites the incorporated fluorophores. The instrument captures 4 distinct images of the flow cell using 4 different wavelength channels corresponding to A, T, G, and C signals. Each base A, T, G, and C is detected in a separate channel within the same cycle image. The emitted fluorescence is separated into 4 wavelength channels and recorded as 4 images per cycle. From this image, the instrument identifies the incorporated base at each cluster position. This is primarily based on the detected color channel, which determines the base identity and is supported by signal intensity, which reflects the strength and the confidence of the signal. The intensity of the fluorescent signal in each wavelength determines the final base call. This process is called base calling. If one strong signal is detected in a single channel, the instrument confidently assigns that base based on the dominant color channel, and the high signal intensity confirms a high quality base call. If multiple weak signals are detected across channels, the instrument compares relative intensities and estimates confidence using statistical modeling. If the signal is too ambiguous, the base is not confidently assigned and may be called as an unknown base N. After imaging, the fluorescent dyes and the blocking groups are chemically cleaved and removed. This restores the 3' hydroxyl group, so the next nucleotide can be added in the next cycle. Then the next sequencing cycle begins. The nucleotides are added again. Incorporation happens again. Washing is performed. Then the imaging and base calling. After that, the cleavage is repeated again. This continues for the total number of cycles in the read. For example, a 150 base read will repeat this process 150 times. Cycle by cycle, the DNA sequence is built one base at a time across millions of clusters simultaneously. As we can see here, nucleotides are being added one after another. This is why it is called sequencing by synthesis, because the sequence is determined as the DNA strand is being synthesized. Here we have discussed 4-channel SBS chemistry. There are other types of SBS chemistries as well, such as 2-channel and 1-channel SBS chemistry. However, we will not discuss those in this lecture. Next, both reads are combined during data analysis. Read 1 provides sequence information from one end and read 2 provides sequence information from the opposite end. This improves alignment accuracy, helps resolve repetitive regions, and increase confidence in mapping DNA fragments. Because both ends of the same DNA fragment are sequenced, this is why it is called pair-end sequencing. Finally, primary data analysis begins. This is where the raw imaging data generated during sequencing is processed into usable sequence information. The software first generates a template map which identifies the location of every cluster on the flow cell. Once the cluster locations are determined, the next step is image processing. The instrument captures fluorescence image during each cycle. These raw images are initially stored as TIFF files. Next, these TIFF images are processed to extract signal intensities. This step converts image data into CIF files. CIF files contain the fluorescence intensity information of each cluster across all channels. Then the base calling is performed. By extracting and using the intensity and the color channel information, the system identifies and determines the most likely base at each cluster position. This converts CIF level intensity data into BCL file. BCL files contain the called bases along with associated quality scores representing the confidence of each base call. The instrument control software also calculates the quality score or Q score for each base call. This Q score represents the confidence level of each base assignment. We will discuss quality score or Q score in details later. Finally, BCL files are demultiplexed and converted into FASTQ files. FASTQ files contain the final sequence reads along with quality scores for each base. These FASTQ files are then used for all downstream bioinformatics analysis such as genome assembly, alignment, taxonomic profiling, or variant detection. In summary, TIFF images are converted into CIF intensity data, then into BCL files containing base calls and quality scores, and finally into FASTQ files containing sequence reads. So the final output is millions of short DNA sequences called reads stored in FASTQ format. Now that we have understood how sequencing works and how reads are generated and analyzed, let's step back and look at the bigger picture. Where is Illumina sequencing actually used and what makes it so powerful? Illumina sequencing by synthesis is widely used in 16S rRNA amplicon sequencing, shotgun metagenomics sequencing, whole genome sequencing, as well as RNA sequencing. It's a powerful tool for studying microbial communities, whole genome, and genetic variation. Now let's look at some of the key advantages. Illumina sequencing offers high accuracy, allowing reliable detection of even small variations. It also provides high throughput, meaning millions of reads can be generated in a single run. And most importantly, it is cost effective on a per base basis. However, like any technology, it also has some limitations. One limitation is the relatively short read length, which can make genome assembly challenging. It also requires complex data analysis and bioinformatics expertise, and in some cases, PCR amplification can introduce bias. So to summarize, Illumina sequencing by synthesis is a powerful and widely used technology. By combining library preparation, cluster generation, and real-time detection, it allows us to decode DNA with remarkable accuracy and scale. This technology has revolutionized genomics and plays a key role in modern microbiology, genomics, and bioinformatics. Today, we have learned about Illumina sequencing in detail, including the complete workflow. So let's bring everything together. Illumina sequencing is a powerful technology that reads DNA one base at a time using a method called sequencing by synthesis. It all starts from library preparation. Then on flow cell, cluster generation creates thousands of copies of each DNA fragment to amplify the signal. Next comes the core step, sequencing by synthesis, where fluorescently labeled nucleotides are added and each base is detected cycle by cycle. We have seen how DNA is read one base at a time and converted into sequence data. These signals are processed through multiple steps from images to base calls and finally into FASTQ reads. In the end, this generates millions of sequencing reads as either single-end or paired-end sequences. These reads form the foundation for all downstream bioinformatics analysis. We have also discussed some key applications of Illumina sequencing. Illumina sequencing is widely used in genomics, microbiome studies, and transcriptomics. We have also explored its advantages along with the limitations of Illumina sequencing. It offers high accuracy, high throughput, and cost efficiency, but it also comes with some limitations like short read length, complex data analysis, and PCR amplification bias. In short, Illumina sequencing transforms DNA into millions of high-quality reads, powering modern bioinformatics and advancing our understanding of life. That's all for today. Thank you everyone.